future doctors. Welcome back to Best Medicine's GCSE AQA Biology lecture series for students in the UK. Today, we'll be starting off with a very important topic, and that is called inheritance. In this topic, we're going to be learning about how genes are inherited from parents to offsprings, and how the physical manifestation of genes comes about. What are the chances that a particular physical manifestation will come about in an offspring from two different parents? For example, you could have a parent with blue eyes and one parent from brown eyes. So what is the probability that the child is going to have, for example, brown eyes or blue eyes, right? So that is inheritance. Before we dive into it, let's have a look at what alleles are. So remember that in sexual reproduction, what you have essentially is a male gamete and a female gamete. And a male gamete contains n number of chromosomes, and a female gamete also contains n number of chromosomes. And the male gamete will have singular chromosomes because it's a gamete, and the female gamete will also have singular chromosomes. So for understanding purposes, let's say this is chromosome number 2 or something, right? And that's also chromosome number 2. So on the chromosome, you could have different genes, right, at different locations. So you'll have gene here, a gene here, a gene here, a gene here, and so on, right? Same for this one. So you have genes here. So let's take this particular gene here in pink and say that this is the hair texture gene, for instance, right? So if this is the hair texture gene, let's say the male parent is giving out straight hair gene, right? So the straight hair gene is coming, or version of the gene's hair texture is straight from the male parent, and a version of the hair texture gene from the female parent is curly. So when we get a zygote, that's going to be a combination of the two. The zygote will have 2 chromosomes, right? One from each parent, and the zygote will contain both the alleles, both the versions of the genes. The zygote will have a straight hair and a curly hair version of a gene, right? These versions of genes, these are called alleles. So a zygote has straight as well as curly, right? Now, this orientation of genes or versions of genes together in a particular zygote is called the genotype, okay? Genotype. The genotype essentially is the genetic orientation of alleles of a particular gene. What is an allele? Allele is 2 or more versions of a gene. So in this case, the gene itself, in this case, is the hair texture gene. What is the hair texture? Is it straight or is it curly? And the versions, we have 2 versions here, so we have straight and we have curly, and those are called alleles, okay? So alleles are versions of a gene. When we jot them down and we represent them as a genotype. So let's say this particular offspring has inherited a straight allele from the male parent and a curly allele from the female parent. And when this person grows up, they end up having straight hair for some reason. Even though all their cells contain both the alleles, they have straight hair. That means that the curly hair allele has been masked off, right? Has been deactivated, and straight hair has been expressed. Straight hair has overcome the curly hair's effects. Straight hair is dominant over curly hair allele. So we have 2 different kinds of alleles. We have dominant alleles, which masks the effect or prevent the recessive allele from being expressed. So in this case, straight hair will be dominant because that's the one that's being expressed, because the child has straight hair only and no curly hairs. And the other one, curly, would be recessive. Okay? Recessive alleles. So we have 2 different kinds of alleles. Dominant alleles that are expressed and mask the effect of the recessive allele. And we have recessive alleles. Now, to be able to represent the alleles, we represent them with letters, okay? With letters. So letters for a particular gene could be any, like A, B, C, or D, etc., right? So let's say this particular gene for hair texture, let's represent this with the letter T. So T is the letter that we use to represent for the hair texture gene. Now, if we're talking about the dominant allele, then we'll write that as capital T. And when we're talking about a recessive allele, then we'll write a small t for it. So capital T represents the allele that has trait here, because trait is dominant. And small t represents curly hair in our example. So dominant is represented by capital letters, and recessive is represented by small letters. So the genotype for this particular individual who has just been formed and born is going to be big T and a small t, right? One dominant and one recessive. And of course, the resulting physical manifestation of this particular genotype is that the person is going to have straight hair. Why? Because they have the capital T. Capital T is the dominant allele that codes for straight hair. The physical manifestation of a genotype, right? For example, this person having the genotype of both big T and small t, and having the physical expression of straight hair, the physical manifestation is called the phenotype, okay? The phenotype. So genotype for the genetic orientation, and phenotype for the physical manifestation of that particular genetic orientation. And that really sums it up. So let's go ahead and review what we have written in our neat notes. So inheritance. In sexually reproducing organisms, every offspring receives half of the DNA from each parent, right? Half chromosomes from this one, half chromosomes from that one. Therefore, resultant genome is a combination of two versions of every gene. For example, a hair color gene could have the blonde version from the mother and a brown version from the father, right? Just like a hair texture gene could have a straight version from a male parent and a curly version from a female parent. These versions of genes are called alleles. So alleles essentially are two or more versions of a gene. Alleles are represented in a genotype, right, by letters. A dominant allele is represented by a capital letter, and a recessive allele is represented by a small letter. Now, for every particular topic in science, we have a pioneer who has done a lot of research and who has come up with these laws and essentially is the founder of this particular science. So, for example, we had evolution previously, and Darwin was a very profound figure in evolution. Similarly, in inheritance, in genetics, we have Mr. Mendel, right? So we're gonna do is we're going to introduce ourselves to Mendelian genetics. Right, so who was Mendel? Mendel essentially was just an Austrian monk, and he was very curious, right? So he was a person who was very, very curious about how inheritance works and how are we able to inherit these traits from our parents to our offsprings, right? And so he had all the time in the world because I guess he was a monk. And what he did was that he grabbed some pea plants and he crossed them with each other to try to observe and figure all of this out, right? Figure out how inheritance works, how are traits inherited from parents to offsprings. So what Mendel essentially did was he grabbed some pure breeding pea plants and crossed them with each other. What is pure breeding? Pure breeding means that we have a particular character trait in an organism, and they have only bred with that particular character trait. For example, you might have heard of pure breeding Labradors or purebred Rottweilers. Similarly, he went ahead and grabbed some pure breeding pea plants. So he came up with these laws of segregation and laws of independent assortment, which we're going to talk about in a bit. And these were able to help us describe how genes essentially are inherited independently of one another. Nowadays, because we have a greater understanding of genes, of chromosomes, where genes are located on chromosomes, meiosis, all those things, we are better able to explain the observances of Mendel and the experiments of Mendel. So what were those observances? What were those experiments? Let's have a look. So here's what Mendel did. He went ahead and grabbed some pure breeding pea plants. Initially, he grabbed some pure breeding purple pea plants, okay? So what I mean by this is that he went ahead and grab some pure breeding pea plants. Let's make a mountain here. Okay, so just for explanation, here's a mountain, right? And then on the right side, we have a field that only produces purple plants, purple flowers of pea plants, okay? We have a field on the right that only produces purple flowers. These are pure breeding purple flower pea plants. They have never, in their history, in their known history at least, produced any other color flowers. So we call them pure breeding because they only have both versions of the genes code for purple flowers. And for on the left side, let's make pure breeding white flowers so that all the flowers in the white one have both the alleles as white flower producing plants. What I mean by this is that if I were to grab a cell from this one, right, both the pairs of the chromosomes on this cell are going to be coding for white. And if I were to grab a cell from these plants, both the chromosomes, every pair would have purple alleles for the hair color, sorry, not the hair color, the petal color gene, right? So that's what he did, and he called them the parent generation. So that's the P generation, the parent generation. And what he did was he crossed them with each other. So he made them sexually reproduce with each other to observe the effects. Now, what he observed was that when he crossed them with each other, pure breeding purple flower pea plants and pure breeding white flower producing pea plants, all the resultant offsprings of his first generation, of that generation that he called the first generation, were purple. All of them were purple. White was nowhere to be seen. All of them were purple. Now, this can be explained now by our knowledge of meiosis and our knowledge of chromosomes and segregation. So let's have a look as to how this works. Remember, the purple flower pea plants, every single one of their cells has two purple alleles, right? A P and a P. And the white ones have a white allele, W and W. Now, before sexual reproduction, they're going to go ahead and make gametes. So when they make gametes, either this one becomes part of the gamete or that one becomes part of the gamete, right? This chromosome becomes a part of the gamete, still codes for P, and that one also codes for purple, and so that one becomes part of the gamete. And this goes ahead and that goes ahead and becomes part of the gamete. Oh, sorry, this should be W and this should be W. Let's illustrate this again here, because it's a little bit confusing over there. So what I mean by this is that when they undergo meiosis, you get gametes, right? And each of those gametes from those individual plants are going to have chromosomes that are singular, not in pairs. And the chromosomes that you get in gametes from the white flower-producing plants are going to have white alleles only. And the purple ones are going to have purple alleles only. Okay? So that is white, white, and that is purple, purple. So when they're going to meet each other and form zygotes, we have different probabilities, right? Different meetups. Either there's a chance that this guy, right, this particular gamete is going to go ahead and meet this gamete and form a zygote. In that case, you're going to have a white allele on the left chromosome and a purple allele on the right chromosome. When this one again, this one goes ahead and meets this, still you're going to get white and purple as the genotype. And this goes ahead and meets this one, you're going to have white and purple. And this goes ahead and gets, you're still getting white and purple genotype. The combination of both white and purple alleles is present, but so that is the genotype. But the physical manifestation of all of these purple-white containing genotype containing flowers is purple only. So why are they only purple? Well, this can only be explained by a notion that purple is dominant over white. Purple is dominant over white. So purple is the dominant allele for the petal color gene, while white is recessive. Now, next thing he did, so all the 100% of the F1 generation are purple. We can also make we call that a Punnett square. And we can make that as well. So let's go ahead and have a look. So a Punnett square is basically you list down the gametes, alleles on the sides, and you find out the genotypes of the offsprings. So our parents were, in the parent generation, we had a PP and a WW. I'm writing W's here just for understanding, but this should be a small p because W is recessive. So PP is going to give a P as a gamete or a P as a gamete. This is going to give you a W as a gamete or a W as a gamete. So everything is going to be PW or WP. And so because P is dominant, all of them are going to be purple, right? Because P is dominant and it masks the effects of white recessive allele. But what happens if you cross this particular genotype, this F1 generation, with each other? Well, if you cross them with each other, what's going to happen is, let's have a look, right? So all the F1 generations, we're crossing them with each other. So F1 generation was PW here and PW here. P and W and P and W. So when we cross them with each other, we get PP, PW, PW, and WW. Right? So in the first one, we have purple alleles, and therefore the resultant phenotype of the flowers is going to be purple. The second one, we have the purple allele, so it's also going to be purple. The third is also going to be purple because we have the purple allele there, and that is the dominant one. But for the fourth offspring here, we don't have any purple alleles present. We only have white alleles. So this is going to be white flower-producing plants. What I mean by this is that if I have a cross between two purple flower-producing plants that have one white allele and one purple allele in their genotype, then you're going to have 1/4 of all the flowers as white. Okay? Let's have a look here and try to explain this. Maybe we can make chromosomes again. So here's a chromosome, and then here's another one. Okay, so we have a purple and we have a white, right? And we have a purple and we have a white. And so that's the P, and that's the W, and that's the P, and that's the W. And so this is going to go ahead and make a gamete with P, this is going to make a gamete with W, this is going to make a gamete with P, that's going to make a gamete with W. And then what we did was we put these gametes and crossed them with each other using the Punnett square. We can just cross them with each other over here as well and get the same genotypes, PP, PW, right? PW, and then eventually WW. Understood? And therefore, he observed that in his second generation, F2 generation, around 25% of all the offsprings were white. This means that white is recessive, purple is dominant, and that genes are being segregated just before fertilization, just before sexual reproduction. The versions of genes are being segregated randomly, and they are randomly joining with each other, and it all works on chance as to what is going to meet what. Okay, and that is called Mendel's law of segregation and independent assortment. Okay, so of course I'm not going to leave you guys here just like that. What we're going to do is another example. So let's have a look. Let's grab some pea plants that have green peas, right? These produce green peas, and some pea plants that produce only yellow peas, right? These are our parent generations. So let's look at the genotypes of the parent generations. And for before we begin, I'm going to tell you that green is dominant, okay? Green is dominant, yellow is recessive. So because this is pea color, we can maybe dictate this as C, right? We can use the letter C for color. So because green is dominant, that's going to be capital C. And yellow is recessive, so that's going to be small c. Because this is a parent generation that is purebred, we will have two C's, both the chromosomes for every cell in the green pea plant are going to have capital C's, and both the chromosomes in the yellow pea plant, as it's a pure breeding generation, parent generation, is going to have 2 small c's. So, what happens when we cross them with each other? Let's have a look. So either this C is going to go ahead and become part of the gamete, or this C is going to become part of the gamete, and for this one, this C or that C, right? And so when they cross with each other, you're going to get capital C, small c for all of them. Capital C, small c. We can also do this in an easy way. We can do it in a Punnett square. A Punnett square essentially is just a square that you make, and you list down the gametes on the top and on the sides for each of the genotypes of the parents, and then you cross them with each other. So for example, the first parent, that is capital C, maybe we can list that here. And the second parent, which is small c, we can list that here. And the resultant genotypes that we get for all the parents is going to be capital C, small c. Now all the resulting parents have the capital C allele, which is the dominant allele, which means that their color is, the peas that they produce are going to be colored green. All of them are going to be producing green peas. So that is our F1 generation. But what if we were to cross the F1 generation with each other? Let's go ahead and cross the F1 generation with each other. So if I were to cross F1 generation, here's what's going to occur. So here's an F1 parent, and here's F1 parent. When we make the gametes, either you're going to have a big C as a gamete or a small c as a gamete, right? And from this one as well, you're going to have a big C and a small c. So when they fertilize, you're either going to get 2 big C's or 1 big C, 1 small c, 1 big C, 1 small c, or 2 small c's. That is the cross between the F1 generation. We can also do this on the Punnett square. Let's have a look. So the parents' gametes are placed on the sides. So we can place this gametes here, and this one's gametes there. So big C, small c, big C, small c. And we're going to get 2 capital C's, 1 capital C, small c, 1 capital C, small c, and small c, small c. And that is very, very unique. Why? Because we have 2 small c's. Okay, so let's have a look at their physical manifestation. The first one, because it contains both the dominant alleles, is going to be green pea producing plant. The second one is also going to be green. This is also going to be green. But the last one, which has 2 recessive alleles, is going to produce yellow peas because it does not have any green dominant green pea producing allele. Okay? Some terms that we're supposed to know now, and you need to pay very close attention to this. When we have 2 alleles that have the same that are the same, we have 2 similar alleles, we call that homozygous. Okay, homozygous. Homo, it means same, and zygous comes from zygote. So this particular zygote has 2 similar alleles, big C and big C. And because this particular one has 2 big C's, we call it homozygous dominant. Okay? Homozygous dominant. And this one here, that's going to be homozygous. Again, why? Because it contains 2 similar alleles. Homozygous, but this is going to be homozygous recessive. Okay? And there we go. What about if we have 2 different alleles? That genotype, what is the name for this one? Here. This is called heterozygous. Okay? This genotype is called heterozygous. The heterozygous genotype is the one in which you have 2 different alleles. A genotype is the genetic orientation of alleles in a particular organism. Their physical manifestation, such as in this case having green or yellow peas as a result, is called the phenotype. So phenotype is the physical manifestation, while the genotype is the genetic orientation. And we have 3 different kinds of genotypes. So let's have a look in your neat notes and review these terms. So genotype. Genotype essentially is the genetic orientation of an organism. For example, a gene for brown hair color, right? A big B, big B, big B, small b, small b, small b. The phenotype, and then we can have different kinds of genotypes. This one is homozygous dominant, heterozygous, and homozygous recessive. Phenotype, the physical expression of a particular genotype. For example, if you have 2 big B's and let's say the dominant is black, so that's going to be black, and heterozygous, big B is black, small b is brown, then it's going to be black. So big B, and small b, big B are going to give you black hair. Homozygous dominant and heterozygous give black hair. If you have homozygous recessive, that is giving brown hair. Dominant allele is an allele that is always expressed if present in a genotype, right? Regardless of if it is present in a homozygous dominant genotype or a heterozygous genotype, this will always be expressed. Recessive allele is an allele which is masked by dominant alleles. It is only expressed when 2 copies of it are present in a genotype, such as in homozygous recessive genotypes. In our example, we had the yellow peas, which were homozygous recessive. So we have these 3 different genotypes: homozygous dominant, homozygous recessive, and heterozygous. Big C, small c, big C, small c here. Okay? And the next topic is going to be sex determination in humans. Now this one is relatively straightforward, but also very, very important. Why? Because you'll be learning diseases that are inherited in a pattern that is sex-linked. So there are diseases that would affect females, for example, or males more, or it's determined by the inheritance of the sex. So remember the sex chromosomes? That is chromosome number what? Sex chromosome is the chromosome number 23, right? So chromosome number 23 can either be X and X, so you'll have a person who's a female, and a female person is going to have an X and an X chromosome pair. A male person is going to have an X and a Y chromosome, isn't it, in all of their cells? The chromosome number 23 in a female is XX, and in a male it's XY. Now, when they reproduce with each other, before reproducing, meiosis occurs. And when meiosis occurs, these chromosomes are split into gametes, right? Are segregated randomly into gametes. You can have this one or that one. So from a female, it's always the X chromosome that comes in, because a female only has X chromosomes to give. But from a male, the male has X and Y. So the male is going to give X as well as the Y chromosome. And now what occurs is that you have fertilization. So either this gamete can go ahead and fertilize with this gamete, or this gamete can fertilize with this gamete, or this gamete can fertilize with that gamete, or this gamete can fertilize with that gamete, giving you XX, XX, or XY, XY as offsprings. So you have 2 females and 2 males here. So there's a 2 out of 4 chance of having a female, 2 out of 4 chance of having a male. And basically what this means is that you have 50% chance of having a male offspring, 50% chance of having a female offspring every single time. We can also make a Punnett square out of this, so let's go ahead and make a Punnett square. We can make a Punnett square, right? So you have a male parent that is XY, and you have a female parent that's XX. So when they make gametes, the X is going to come here, the Y is going to come here, the X is going to be here, and the X is going to be here. And so you're going to get 2 females and 2 males in a pool of 4. And that gives you a 50% chance of males, 50% chance of females. Okay? Very, very simple. This is quite simple, but it's inheritance that is linked to diseases, which is very important, is a little bit tricky. But before we go into this, I wanted to bring another topic to your light. It's a little bit extra, but it's very important information for general knowledge as well. And that is the blood groups inheritance. Okay, the blood groups inheritance. The blood groups inheritance is a little bit unique. because it works on a system of codominance. Codominance because we have two dominant alleles in blood groups. We actually have three alleles for blood groups. So blood group essentially is an antigen that is present on the red blood cell. Remember red blood cells, right? The red blood cells have these antigens on them. Okay? You could either have an A antigen, you could either have a B antigen, or you can have no antigen at all, right? These are three different kinds of alleles that we have. So you have antigen A, so I is the gene dictator, and then this is the allele dictator. For the blood group, the blood group can be B, which is dominant, or small I, that is recessive, and that is essentially blood group O. O is recessive. So what I mean by this is that if a person has A blood group, so let's say a person has A blood group. If there's a person who has A blood group, they're going to have either this IA in their genotypes, right? Both the chromosomes are going to code for A, or they can have A and O, because this is recessive, so this will be masked by the dominant A allele, and so therefore you're going to get a phenotype of A blood group. For B blood group, a similar thing would occur. For B blood group, you're going to have IB, right? B, or B blood group and an O allele. O is recessive, so it will be masked off, and both of these together are going to give you a B blood group. For O blood group, which is a bit rare because it is recessive, you need to have two recessive alleles, homozygous recessive, to have an O blood group. Okay? If you want to learn about the ability to be able to donate or to be able to receive blood, that is a little bit extra, okay? I'm not going to teach it to you over here, but you can send us a message if you want to learn that extra stuff as well. That is medically important as well as important for day-to-day knowledge. But I would encourage all of you to go ahead and research how do blood groups donations, transfusions essentially work. I also have a question for you guys. So which blood group is referred to as the universal donor and which the universal acceptor? And please answer in the comments, and please mention your nicknames, please, okay? And yeah, next up we have family trees. Alright, so this family tree thing is very, very easy. It's so easy that I haven't really written an explanation for this because it's so self-explanatory. Basically what you have is a male that is denoted by a square and a female that is denoted by a circle. At least for the GCSE AQA levels, you only have these. And a shaded the box means an affected person or having the phenotype of a particular genotype that we're talking about, while the unaffected does not have the physical manifestation of the gene that we're talking about. This could be a disease, for example, right? Like cystic fibrosis. So this person here is a male, over here is a male, and has the disease, and this person is a female who has the disease, and so on. So we'll come back to this in a bit and try to make sense of this after we do some diseases. Now, diseases can be of two types, right? Genetic diseases can be of two types. Genetic diseases can either be dominant or recessive. So we will have two patterns of inheritance. And secondly, genetic diseases can either be present on autosomes, remember autosomes, chromosome 1 to 22, or genetic diseases can be present on sex chromosomes, especially the X chromosome, okay? So genetic diseases are either autosomal, dominant or recessive, or X-linked, not sex-linked, X-linked, dominant or recessive. Why just X-linked? Why not Y-linked? Because X chromosome is very big, right? Y is very, very small over here, right? That's a Y chromosome doesn't have much of a gene there, so it doesn't have too many genes, and therefore it doesn't really carry that many genes, so we don't have that many mutations over here, and we don't have too many genes that we need to know. But for X chromosome, it's a very big chromosome. It has like 900 genes, and so very easy to get a lot of different diseases that are linked to the X chromosomes, just because it has so many genes, and so more chances of different kinds of mutation that could occur on that chromosome. So X-linked recessive or dominant diseases exist, and autosomal recessive and dominant diseases exist. The first disease that we're going to do is polydactyly, or polydactyly, okay? That is very easy. Basically, what you have in this disease is that you have joined fingers or an extra finger. Okay, so offsprings have an extra or joined fingers. And this is an autosomal dominant disease. Okay, autosomal dominant disease. What this means is that if I were to sort of denote the disease gene by a D, then the capital D codes for disease and small d codes for normal. So if a person has two capital Ds, they are affected, right? Even if they have a capital D and a small d, they are heterozygous, they are still affected. But if they have two small d's in their genotype, homozygous recessive, then they are normal. Only then they are normal. So that is how dominant diseases work. So let's do a couple of questions for this. By the way, dominant diseases will be present in every generation. You'll understand this in a bit. But let's do some questions on this. So here, if a parent with polydactyly, heterozygous, so we have a heterozygous parent, marries a normal individual, what is the probability of their child having polydactyly? Well, whenever you see a question like this, the first thing you want to do is write down the genotypes of the parents. Write down the genotypes of the parents, and then do the Punnett square. So write down the genotypes of the parents, and then do the Punnett square. So here's what we're going to do. Genotype of the first parent. If a parent with polydactyly marries a normal individual, and they're saying he's heterozygous or she. So that's going to be big D, small d, if we use the letter D. Normal individual. Because polydactyly is a dominant disease, a normal individual should be homozygous recessive. And so two big heterozygous here and two small d's here. And now we just do a cross. So let's go ahead and Punnett square this out. So we get big D, small d from this parent, heterozygous, and two small d's from the other parent. And we get two heterozygous offsprings out of 4, and two homozygous recessive offsprings out of 4. So these are heterozygous, and they have the disease allele, so these are affected. And the other ones are not affected. So 2 out of 4, which is 50% of the children, are going to have polydactyly, okay? They're going to be affected. Now the next question is, if both parents are heterozygous for polydactyly, what is the probability of the child being normal? So again, the first thing you do is you write down the genotypes of the parents. So as both parents are heterozygous, we're going to go ahead and write D and D, capital D small d, and capital D small d. And then go ahead and do a mono hybrid cross Punnett square for them. So capital D small d, and we get two capital D's, one capital D small d, and two small d's. So in this case, we get a homozygous dominant, heterozygous, and a heterozygous. All of these are affected. Why? Because again, polydactyly is a dominant disease. So as long as you have the dominant allele, you're going to have the disease. This one here is unaffected. That's normal. So 1 out of 4 is normal, or 25% children are going to be normal in this particular set of parents. Next question is, if a parent with polydactyly, homozygous dominant, marries a normal individual, what is the probability that their child is going to have polydactyly? Well, let's have a look. So again, the first thing you do is write down the genotypes of the parents. So if a parent with polydactyly is homozygous dominant, they're saying, The parent is homozygous dominant. Capital D, marries a normal individual. Now, normal individual is supposed to be homozygous recessive because polydactyly is a homozygous, a dominant disease. So here's a normal parent, here's a homozygous dominant parent, and we do a Punnett square for them. So capital D, small d, and all the resulting individuals are going to be heterozygous. So because all of them are heterozygous and it's a dominant disease, all of them are going to be affected. Okay? So the answer to this is 100%. Or 1. Okay? Next up, we have cystic fibrosis. Cystic fibrosis is an autosomal recessive disease. So unlike polydactyly, which is dominant, this is autosomal recessive disease. Essentially, what occurs in cystic fibrosis is that the person who is affected with it gets thick mucus secretions. What is mucus? Do you remember in the respiratory system chapter, we looked at the trachea and the bronchi, and we said that the bronchi and the trachea contain cells in their epithelium called goblet cells, and those goblet cells were producing mucus. And the respiratory cells, the respiratory epithelial cell, had cilia, kind of like this, that you have these cells, right, that have cilia on them, and you have another group of cells that is producing all this mucus, right? And ciliated cells are removing the mucus, and these are called goblet cells. So in this disease, what happens is that you have a very thick secretion of this mucus, very thick mucus being produced here, and it is very difficult to be removed, which means that the airway gets blocked and all of this junk sort of accumulates here because it's not being removed. So here's a comparison of a healthy bronchus and a bronchus with a person with cystic fibrosis. So healthy bronchus is very clear, right? Very open. There's nothing in it. Very good airflow. But for a person with cystic fibrosis, they have mucus clogging, right? So mucus clogging results in blockage of the airway, which means that there's a poor airflow and there is the piling up of all of this bacteria here, which causes recurrent respiratory tract infections. So you have all this junk accumulating here, all the bacteria and everything, and so because it's all piling up, it increases the risk of infections, leading to recurrent respiratory tract infections. Okay, and that is cystic fibrosis. So that is an autosomal recessive disease. Now, the way this would be inherited is a little bit different than polydactyly. So let's have a look. Maybe we can use the letter C for cystic fibrosis. So capital C is going to be normal, and small c is going to be the diseased allele. Why? Because it's a recessive disease, right? So capital C and small c. Let's have a look. So a person with two capital C's, that is homozygous dominant for normal. So this person is normal, right? What about this one here? Capital C, small c. Well, that is heterozygous. Now this person does not have the disease, but they do carry the allele for it. So we call such a person a carrier, okay? So a person who is not affected with the disease, but they have the allele for it. And that is called a carrier. A heterozygous individual for an autosomal recessive disease is called a carrier. And of course, then we'll have the homozygous recessive. And in homozygous recessive, you do have the disease, right? Because it's a recessive disease. Both affected alleles are there. Okay, now if I were to represent this information on sort of like a family tree, then here's what I would do. I would go ahead and draw a parent. Let's say here's a parent who's a male, right? And here's a female parent. Now if I want to make the male parent as homozygous dominant, then they would have a clear. Let's say here's a homozygous dominant parent, right? Very clear square box. An affected female is going to have a completely shaded box. Okay? So that's a homozygous recessive female. And then they can have children. And all of those children, as you can see, you can do a Punnett square here. And what you're going to get is all the heterozygous individuals. Right? And you get multiple children, let's say you get a couple of males and a couple of females. So here's a female, here's a male, and here's a female, and here's a male. 50% ratio. All of them are going to be carriers, right? So this male is going to be a carrier, and this female is going to be a carrier, and we denote carriers as half shaded because all of them are heterozygous, okay? And so on. So that's how we worked with family trees. So maybe we can look at the family tree that we saw earlier and work with this. Okay, so let's assume that we're talking about cystic fibrosis. If we're talking about cystic fibrosis, then this one here, even though they've given us the carrier genotype, it's an autosomal recessive disease, because this person is affected, but this person is not. This is a carrier, so this should be half shaded. Okay, but anyways, a carrier versus a carrier mating an affected person, and you get a 50% probability of having the disease. But here's the resultant genotypes of the children and so on, right? And then this is mating with this one and so on. So that's how family trees work. Now let's do a couple of questions on autosomal recessive diseases. So the question is, if a carrier parent, heterozygous, marries a normal individual, what is the probability of their child having cystic fibrosis? Well, it's very easy. The first thing you do is you write the genotypes of the parents, right? They're saying one parent is the carrier, so capital C, small c. You can assume any random letters if they don't give you letters. I'm using C here, but you can use anything. Marries a normal individual. So a normal individual, phenotypically, a normal individual can either be a carrier or homozygous dominant. But in this case, I'm just going to assume, because I made this question and I haven't worded it, normal homozygous individual. Homozygous individual who's normal, okay? So that's going to be homozygous dominant. That's CC. So when we cross this, what we're going to get is, so what is the probability of the child having cystic fibrosis? Let's have a look. 2 capital C's, 1 capital C, 1 small c, and the resultant genotypes that we get is this here. So we have 2 homozygous dominant and 2 heterozygous. Do we have any homozygous recessive? No. So because we don't have any homozygous recessives, we don't have anyone affected with the disease. So the answer to this is 0%. What about this one? If both parents are carriers for cystic fibrosis, so both parents are carriers, right? That's our genotypes. What is the probability of the child having cystic fibrosis? So let's have a look, right? Let's write down the genotypes of the parents first. So one of the parents is going to be capital C, small c, and the other parent is also going to be capital C, small c. And what we get here is big C, small c, big C, small c, and we cross them together. Big C, big C, big C, small c, big C, small c, and 2 small c's over here. That's 1/4, right? 1/4, or 25% chance that the child is going to be affected with cystic fibrosis, okay? Question is, if one parent has cystic fibrosis and the other parent is a carrier, what is the probability of their child having cystic fibrosis? Let's try to do this. So this is very easy. What you do is you write down the genotypes of the parents again. Two small c's. One of the parent is a carrier. 1 big C, 1 small c. Let's go ahead. So small c, big C, small c. And that's going to be... This is diseased. These are carriers. So disease is 50%, 2 out of 4, right? And the answer to this is 50%. Now, fortunately for AQA at least, there are no diseases that you need to know that are linked with the X chromosome. In other boards, you are supposed to know X-linked recessive and dominant diseases. So if anyone who's doing maybe EdXL or CAIEs, if you're watching this, you might want to review this from your own resources, or you can send us a message and we can review it together, okay? Down in the WhatsApp link below. But for you guys, for AQA students, you're not supposed to know the X-linked dominant or recessive diseases. That's a little bit complicated, and all you need to know is autosomal dominant and recessive diseases. So next up we have genetic engineering, which is the last topic of this particular lecture. Genetic engineering involves altering an organism's DNA to change its traits. So what is genetic engineering? You know what engineering is? Engineering is being able to change things to improve stuff, right? Towards betterment. So in this case, what we're trying to really alter is the genes. We're trying to alter the DNA to try to do a couple of things. We can do genetic engineering to do something called gene therapy, which helps essentially treat diseases. We can also produce essential medicines like insulin, and we can also develop advanced treatments such as something called CAR T cell therapy for cancer. Okay, now we have talked a little bit about this earlier as well, so I'm just going to go ahead and explain to you how you would make insulin essentially from genetic engineering very quickly, okay? So remember that in type 1 diabetes, right, type 1 diabetes mellitus, you're not able to make your own insulin. So you're not able to make your own insulin. Maybe you can take that insulin gene and put it in something that can, right? So here's what we do. We take a cell from a person, right? Here's a cell, and that cell has nucleus, and in the nucleus you would have the DNA, right? And in the DNA you would have the gene for insulin. So here's the gene for insulin. So again, we've already talked about this once, but I'm just going to very quickly review this for you. So what you do here is you take that particular portion of DNA that contains the gene, so let's say here's a gene, and you cut it using enzymes called restriction enzymes, right? We've already gone over what restriction enzymes are. So you cut the gene, and now you have the gene, right? You have the portion of DNA here that contains insulin. Now what you do is you take the gene and you insert it in a bacteria. So here's a bacteria, and the bacteria would have its own DNA. So here's the bacterial DNA, let's say, right? Double-stranded DNA of the bacteria. And what you do is you take the restriction enzymes and you cleave it here, right? Cleave it here, make some space, and insert this into the bacterial DNA. And now what you get as a result is a bacteria that has been modified. Its DNA contains its normal DNA on top of the insulin-containing gene as well. So now when the bacteria is going to go ahead and make its own proteins, right, using the gene, using transcription and translation, it's going to make its normal proteins as well as it's going to make insulin. Okay? Now next thing you do is once you have this bacteria, you just culture it. You culture this bacteria and grow many colonies of it in the industry. And this is going to keep on making insulin over and over again, and you keep on extracting that insulin, and you can use it in insulin pumps to treat diabetes mellitus, right? That's a very good example of genetic engineering to play with genes to bring about beneficial changes. Now, something very important, genetic screening. So genetic screening is the process of looking at cells or chromosomes or DNA for genetic abnormalities. Like, for example, we have mutations that we've just studied. For example, cystic fibrosis is a genetic disease, right? We can look at the DNA sequence to try to see if there's mutations for cystic fibrosis. Polydactyly is a disease, and we can look at genetic mutations for polydactyly. Similarly, Down syndrome is a very popular genetic anomaly, and we can look at chromosome numbers to try to see Down syndrome. Down syndrome actually is described as trisomy 21. This is not an AQA term, but you should know, I guess, for general information, that it's called trisomy 21. Trisomy 21 basically means that normally you have two pairs of chromosome number 21, right? We have 23 chromosomes, 1 to 23, and we also have a pair of 21 chromosomes. But in Down syndrome, there's 3 of them, okay? And that's basically Down syndrome. So you can take a cell and then freeze it maybe in the metaphase and look at it with a microscope and try to see if it has 3 21 number chromosomes or not. So that's the Down syndrome. So that's genetic screening. Genetic screening basically is when you look at cells, DNA, or sequence for genetic anomalies. So how do you obtain these cells? How do you get these cells? Well, there's 2 ways. One of them is that you can take it from the placenta directly, right? From a unborn baby, you can take it from the placenta. Or the other is that you can take it from something called the amniotic fluid. You know this fluid in which the baby is sitting and swimming before it's born, it's being nurtured? That's called the amniotic fluid. And so you can take the amniotic fluid of the body, of the baby, and that contains a bit of cells, and you can examine these cells. Or you can directly take a piece of the placenta. Okay, so let's have a look. So for example, if this is the belly of a mother, and here's the baby, right? And that's a very big belly. Maybe we can read about this. Okay, so here's the baby, and then that's the placenta over here. We can take some cells out of this, right? Extract some cells out of this by a needle and examine those cells. The other thing that we can do is this baby is swimming actually in a fluid, and this fluid is called the amniotic fluid. So we can also insert a syringe into this one and take some fluid out, and the fluid will contain some cells and we can observe them. Okay, so these are two ways. The one on the right, in which we take the amniotic fluid out, that is called amniocentesis, okay, or amniocentesis, in which we take some of the amniotic fluid out, which contains cells. The other one is called chorionic villus sampling. Chorionic villi essentially are just components of the placenta. So that's what we do. We take some pieces of placenta out and examine those cells. So chorionic villus sampling is when you take portions out of the placenta and look at the cells. And amniocentesis is when you take out the amniotic fluid and look at the cells there. And then again, these can help us diagnose conditions such as Down syndrome and cystic fibrosis, etc. Okay? Now, there are concerns about genetic screening. This is an invasive procedure. Invasive means that you are invading the body tissues physically. So there is a slight risk of miscarriage and birth defects because of these invasive testing procedures. There's also ethical concerns about selecting embryos based on genetic traits because, for example, somebody has a genetic disease and people might want to abort it, and then there's ethical concerns about whether this should be allowed or not, right? Abortion should be allowed or not if somebody, let's say, has a genetic disease. Potential for discrimination. Again, it's the same thing. Potential for discrimination means that we have individuals with genetic disorders and these can get aborted. These testing facilities are also have high costs, and there's also emotional impact on parents who face difficult decisions. There's also privacy issues regarding genetic information. So these are some of the concerns. Not very high yield, but you should know about these, okay? CVS and amniocentesis, what is genetic screening, and what is genetic engineering, all right? And yeah, that pretty much sums up this particular lecture. So hopefully you guys can do a lot of practice for this particular lecture's contents and inheritance, especially, right? Asexual sexual reproduction and so on. And if you're having trouble solving those questions, if you're having trouble getting familiar with the mark scheme, just let us know down at the WhatsApp link below, okay? And I'll see you guys in the next one.